So this short video is all about how gene expression is controlled within cells. Right, there are four main ways that gene expression can be controlled. Transcriptional, so genes can either be turned on or off. Post-transcriptional, so messenger RNA can be modified, which regulates translation of the different types of proteins that are produced. Translational, so translation can be stopped or started. And post-translational, which involves proteins which are modified after synthesis, which changes their function. Right, so the first method that we want to look at is transcriptional control. So most mechanisms that control gene expressions do so by controlling transcription, so the synthesis of messenger RNA. Right, chromatin remodeling is the dynamic modification of chromatin architecture to allow access of condensed genomic DNA to regulatory transcription machinery proteins and thereby control the genes. Right, the following short video hopefully summarises all of the key points to go with this.
Transcriptional control is all to do with either promoting the transcription of messenger RNA by DNA polymerase or repressing it. Right, so this can be done in a few different ways. So repressed proteins or complexes of repressor proteins can bind onto the DNA. Right, if a repressor protein is there, that physically goes and stops the RNA polymerase from going and transcribing it. You can have promoter proteins or promoter factors that go and bind onto the DNA, which causes the RNA polymerase to bind to it more strongly, therefore you get more messenger RNA being produced. Both of those things that are like added onto it we call epigenetic changes, right? Because we're physically attaching stuff to the DNA. Right, on the DNA itself, you'll have areas called promoter regions, which are also called Tata regions because they've got the nucleotide sequence TA, TT, AA, which physically promote the transcription of the gene that comes after that region.
The second way that we want to look at of how the physical DNA can be changed is to look at heterochromatin. Right, so this is tightly wound DNA causing chromosomes to be visible during cell division. The transcription of these genes is not possible when the DNA is tightly wound because RNA polymerase cannot access the genes. Protein synthesis does not occur during cell division, so it only occurs during interphase between divisions. Right, this is a simple form of regulation that ensures the proteins necessary for cell division is synthesized in time. It also prevents complexes forming and energy being consumed during the process of cell division. Right, the next short video will go and summarize the key points about this.
The second type of gene control is when the DNA is wrapped around heterochromatin, right, or wrapped around this A protein complex to form heterochromatin. Right, this happens during mitosis and it causes the chromosomes to condense. During mitosis, we don't want any of the genes to be transcribed as the DNA could become damaged and all of the energy that the cell's using needs to be used physically for mitosis, not to go and carry out any protein synthesis. So the DNA gets tightly wound this to form heterochromatin. The protein will have a slight positive charge because DNA is slightly negatively charged. That's what binds it to it. So the DNA gets tightly wound around it, which means that the RNA polymerase can no longer bind to the DNA because it's not physically got enough room to move around, so therefore no messenger RNA can be transcribed.
Next thing that we want to look at is histone modification. So DNA coils around histones because they are positively charged and the DNA is negatively charged. Histones can be modified to increase or decrease the degree of packing by the addition of acetyl group or phosphate groups that reduce the positive charge on the histones, which causes the DNA to coil less tightly, allowing certain genes to be transcribed. However, the addition of methyl groups make the histones more hydrophobic, so they bind more tightly to the DNA, preventing transcription. The next short modelling video will show how this happens.
So the third type of gene control is to do with histone modification. So DNA is wrapped around histone complexes, which are made up of 8 proteins that can be modified to either make them less positive or more positive. Right? So these histone complexes are slightly positive because DNA is slightly negative, so therefore the DNA will bind to it. If acetyl groups are added to the histone complex, that makes it less positive, so therefore the DNA binds less tightly to it and can be transcribed by RNA polymerase. If, however, these histone groups are modified by adding methyl groups to it, that makes them more positive, therefore the DNA binds more tightly, so the RNA polymerase, well, it can't get around it, so it can no longer transcribe the genes on that DNA.
So, genes and DNA sequences that code for a protein come in long strands that are made of extrons and introns. Right? The coding region of a gene are called extrons, and the intervening non-coding regions are called introns. After transcription of the DNA into primary messenger RNA, the non-coding parts are spliced out to go and make a mature strand of messenger RNA that can then be used in protein synthesis. My following short clip will show how this is done.
So, RNA processing happens after the messenger RNA has been transcribed from the DNA. The DNA contains extrons and introns. Remember, only the introns go and code for genes. After the RNA has been transcribed into messenger RNA, it's then edited. Right? Sometimes this is called RNA splicing because parts of the unedited messenger RNA is spliced out. So we end up with edited messenger RNA that now only has the code for the particular protein, and at either end we go and put a cap just to go and hold it together.
Another way that gene transcription can be controlled is by transcriptional control. Right, there are 3 main ways that this happens. Either by the degradation of messenger RNA, so the more resilient the molecule, the longer it will last, so the greater the amount of translation. Binding of inhibitory proteins to the messenger RNA that prevents it binding to the ribosomes, and activation of initiation factors which aid the binding of messenger RNA to the ribosomes, so therefore more transcription and translation even will happen, so more proteins will be synthesized. In post-transcriptional control, it's all about how the physical proteins are changed. Right, this can be done in 4 main ways. So the addition of non-protein groups such as carboxyl chains or carbohydrate chains, lipids or phosphates, modifying amino acids and the formation of bonds such as disulfide bridges, folding or shortening proteins, and finally modification by cyclic AMP. So for example, in the lac operon, cyclic AMP binds to the cyclic AMP receptor protein, increasing the rate of transcription of the structural genes. Hopefully this video should have helped you complete your daily revision task. Please subscribe so you can keep up to date with other videos to help with your daily revision tasks. You can follow me on Facebook or follow me on Twitter to help you keep up to date.